My name is Patrick Ettenhuber. I'm the product owner of the QIAGEN CLC Microbial Genomics module, which will be the main module that is used throughout this session. I will go into more details about which products we have available for microbial and microbiome data analysis in the following. Let me first start by giving our standard legal disclaimer. The QIAGEN products shown here are intended for molecular biology applications. These products are not intended for the diagnosis, prevention, or treatment of disease. For up-to-date licensing information and product-specific disclaimers, see the respective QIAGEN kit handbook or user manual. The handbooks or user manuals are available on www.qiagen.com or can be requested from QIAGEN Technical Services or your local distributor. So today's session will mark the end point of the Human Microbiome Days presentations. We have seen presentations on sample disruption, nucleic acid isolation and automation, enrichment and amplification, PCR digital PCR and NGS, and today will be solely about data analysis, specifically about the data analysis that comes out of NGS sequencing machines. During the Microbiome Day session of the bioinformatics analysis, I have asked you a question about which bioinformatic challenges you would like to see tackled in today's session. And you have answered that you would mostly like to see statistical analysis of metagenomic data. This is also what I'm going to focus on today, but I will also try and see how far I can get into metagenome assembly, whole genome alignments, and in the end, I will also briefly scratch the surface of the taxonomic analysis of 16S ITS, since it's very similar to what we will also be doing today. Let me briefly reiterate on which product we're talking about. This is the QIAGEN CLC Genomics Workbench Premium package, which includes a number of modules. The main module, as I have mentioned previously, is the QIAGEN CLC Microbial Genomics module for the analysis of microbial NGS data and metagenomic NGS data. In the bottom of this of the slide, you see also the links to the products. You can also have it a little simpler if you want to. You could just start by Googling CLC Genomics Workbench Premium, and you see I've already done so. And going to this page lets you actually request a quote, and you can get 14 days of analysis for free. So if you're interested, please go to the page and request your quote here or a trial. This is including the modules I've been talking about. If you more specifically look for information on the CLC Microbial Genomics module, you would use your favorite search engine and search for CLC Microbial Genomics module. On that page, you find a lot of information what the module is for. And specifically here, I would like to point you to the manuals on how to use it, but even more important to get started are the resources. And under the resources, I would really like to highlight the tutorials, because they are really step-by-step tutorials for beginners if you're not used to working with CLC yet. There are video tutorials and sort of describing how our algorithms perform. We have benchmarks, application notes, and white papers also listed. Yes, and then it's already time for the live demo. So this is the CLC Genomics Workbench 21.0.4. This also what you can see here is the standard welcome screen. It already presents you with some possibilities where you can choose and select to, for example, import data, get started, open the welcome center. There are direct links to different plugins and information which we think that are necessary or interesting for you to read. For example, also tutorial data is listed here. But in general, this space will be occupied by whenever you open a data element, sort of the visualizations of your data. In the upper left, so in the top row, you find in the upper left top row you find different buttons which you can use to import, export data, launch tools which you would like to launch, cut, copy, paste, delete, sort of standard controls. Below that is the navigation area where your data elements are stored in a typical folder structure, and you would be able to to structure it in the way you like to. Below the navigation area, you will find the toolbox. Note also that the toolbox again reappears in the very top of the screen as one option. This under the toolbox pane here or in the top, you will find all the tools that are available for you. Some of the tools will only become available upon installing a specific plugin. For example, this Microbial Genomics folder here, that will only appear when you have installed the Microbial Genomics module. In order to install modules, I would like to point you to the upper right corner of the workbench, where you can either select to go to your workspace. There's a plugins button, a references button, and some download buttons. We'll start by looking into the plugins button because you have requested that you would be interested in seeing how whole genome alignments are performed. For this, we also have a specific plugin. So you would open this plugins, download plugin, find the whole genome alignment plugin, click on download and install, accept the terms, and close and restart the workbench. And then you will see that there will be a new folder in the toolbox that is called whole genome alignment. So this is the folder, and it comes along with some tools. We'll have a brief look into those later. Let me now present you with the data we are going to analyze today. We will be looking into a paper that is called Genome Sequencing of Sewage Detects Regionally Prevalent SARS-CoV-2 Variants. So this is not so much a human microbiome analysis as it's a human associated microbiome we are looking at. Specifically, it's this is about viral enriched reads or reads that have been enriched for viral sequences, but it's still a metagenomic sample, and it's a mixture between stool and everything else that ends up in the sewage. So the way to analyze this kind of data is very similar to how you would analyze human microbiome data. The difference often is that in the human microbiome data, you would expect many human reads depending on how you did your sequencing. So you would try to remove those during the analysis. We're not going to do this here because of the enrichment process. Most of the reads are already or many of the reads are already viral reads, and we'll only focus on that part. So to find the respective data, let's see if there is a bioproject number in here, and there is. So we would like to download the data that is associated with that paper, and we can do so by copy pasting this bioproject number, And then we can use the next button up here that is called download. Here you have several options. We'll choose search for reads in SRA. We'll just look for the bio project because we know it's a bio project number. It's a little bit quicker if we only search in that column. Click enter, and then you see a bunch of lines appears. If we can now inspect the metadata or select them all and choose to download reads and metadata. I've already done that because the download takes some time. What you will then get is a couple of reads files and some metadata that is associated with those reads. The interesting thing now is that the reads elements are associated with this metadata such that we can always analyze our data in the context of this metadata. One problem here is that some of the metadata does not come directly with the metadata that is stored in SRA, but some metadata is actually only available as an Excel spreadsheet that lies under the that is only available from the journal's homepage. I've also already downloaded that and stored it into my on my machine. Well, in this spreadsheet, I have associated the run accession numbers with the sample names that are coming from SRA, which is basically this library name here and the run accession number. So in this way, these two metadata tables can be joined. We'll use that later. So a typical analysis starts out by cleaning up the reads in some way. So we would like to trim the reads, remove adapters, and things like that. In order to do that, we can use a tool that is called trim reads. You can either navigate to it in the very large and comprehensive toolbox, or my favorite way is to actually search for it, because then it will filter the large list of tools down to what is interesting. So if I search for trim reads here, then I have only a few options left, and this is the tool we are looking for, trim reads. In this case, a wizard will open, and then we can select the reads we want to. We can either do so by adding them individually, or we can right-click on the reads folder and say add folder contents. We'll choose next, keep all as is. We'll just believe that there are reasonable standards set in the wizard for now. And then the last step lets you either open or select some output options, or it lets you save or open the result. We'll choose save and make a new folder with the trimmed reads and click on finish. And this is the folder where our reads will be stored into. If we now go to the processes button, tab in the toolbox pane down here, you can actually see that we now have an active process running. So while this is, of course, I have already done some analysis before, because otherwise we would run out of time here. So I have already trimmed the reads. The output would look then very similar. You would get all of your reads files again with the trim pairs and a trim report, which I have chosen to save here as well. If you open this report, there will be a whole lot of information about your samples, the number of reads, average lengths, how many reads were trimmed, and how long the reads were after trimming, and a lot more statistical information on that. We'll not go into details here. We just note for now that these reads are now fit for running a taxonomic profiling with it, which is the whole metagenomic taxonomic profiling, and that is a tool that is available with the Microbial Genomics module. Let's have a look at what is available in the Microbial Genomics module. So here we can see this is the folder structure of the Microbial Genomics module. We have tools for metagenomics, typing in epidemiology, for isolate analysis mostly, functional analysis and functional annotations of genomes, and metagenomic functional profiling. We have drug resistance analysis, and a folder that lets you download the necessary databases that are required for building databases that to analyze metagenomics or typing data. So for the taxonomic profiling tool is of course to be found under metagenomics, and there we can have two different ways of analyzing metagenomic data, that is either amplicon based or taxonomic analysis. In this case it's not amplicon based but whole metagenome sequencing, or targeted metagenomic sequencing rather. Furthermore, there is an abundance analysis folder, which we'll look later into, which contains all the tools for statistical analysis of metagenomic data. So, the taxonomic profiling tool can be found here. And once we have selected the taxonomic profiling tool, we can basically use our trim reads in the same way as before. Add folder contents. Well, no. It says select exactly one element. It only lets you analyze one sample at a time, but there is also a solution for that. We can choose to run it in batch mode. That means basically run this tool one sample at a time, and then we try again, add folder contents, and then it will allow you to add all of these. In the next step, it shows you the batch overview, all of the individual steps that will be run. And then you will have to use a database. Well, now I have already pre-selected a database that is called Clustered Reference Viral Database. A brief note on that: this is a database that is supplied from the FDA, RVDB, and can be found under this rvdb.dbi.udel.edu, which is the official homepage for the RVDB, but we have made it a little bit more simple for you. You can actually also get the database from within the CLC Genomics Workbench, navigating to the databases folder, which has the same structure as above, taxonomic analysis, and then you can download a curated microbial reference database, and there you can see that we offer a range of options. The QIAGEN Microbial Insights prokaryotic taxonomy database for prokaryotic analysis. In this case, it would be viral analysis, and you can choose the clustered reference viral database here. This is what I did: downloaded it as a taxonomic profiling index and stored it in my navigation area. So again, let us run the taxonomic profiling tool. And why is it we do that? First of all, we would like to get an overview on what is in those respective read sets, sort of which viral species are represented. But the taxonomic profiling tool does more. It also bins your reads. It associates a taxonomy with each individual read. And we can grab those reads later and analyze a specific subset of reads we may be interested in. So we can choose to use we would like to produce an abundance table. We would like to produce reads that match the reference database. We could, in principle, also choose to save unclassified reads. We're not going to do that, but we will always get a report in order to see what the algorithm has been doing. Then we can choose to save in a specified location and create a one subfolder per batch unit that is per sample. It will create all of these files, an abundance table, reads matching the reference database, and a report. Click next. And then again, we would make a folder and choose to save our results in that folder. Now this analysis takes a little bit of time. You can see again it's running, but I will not wait for this to be completed. I would rather look into the pre-calculated results, so it will look like this once it's done, where you have one folder per read file, and now we have the database matches stored in that folder, an abundance table that tells me what's in those samples, and a report. Let's briefly open the report. It's a very simple report. It will tell you how many different classifications it came up with on each of the taxonomic levels. So how many different species it has found. It says 43. It found 31 genera and so on. With respect, it also gives some classification of reads. You can see this sample here, for example, only 4,000 viral reads have been detected. Since we used a viral reference database, the 77,000 here could not be classified. I have run the taxonomic profiling tool again on those in a sort of to look into what is that, and many of those reads are actually bacterial reads. But since we are interested in detecting SARS-CoV-2, here we are only interested in the ones that actually match our viral reference database, and here it's only 4,000, which is not too many. But let's have a look into the into how an abundance table looks like. An abundance table is basically a table with nucleotide references or assembly references on the very left that have an associated taxonomy, and some reads that have been assigned to it. And here you can see that most of the reads have been assigned to tomato brown rugose fruit virus, which is in accordance with the paper, by the way. And still many reads were associated with the severe acute respiratory syndrome related coronavirus. In all, you can see it occurs several times, which means there are several references in our reference database that have been hit by those reads. So different strains, if you want to. We can get an overview over the species by aggregating the abundance table on a specific level of the taxonomy. And then we can look into the species level. And here you can then see the aggregated number for the severe acute SARS-CoV-2. Here, 800 reads is not too much. We don't expect that we can do a lot with those, so we'll have to look into a different sample as well. I have previously been looking into this 170 here. Now we'll do the same trick again. We'll collect on species level, sort the abundance table, and then we can see, okay, there's something else, private virus, which is the most abundant one, but here we get almost 5,000 reads associated with SARS-CoV-2. Since we selected to store those database matches here, we can just select the respective SARS-CoV-2 line and say extract reads from selection. And then it will open a new data element with exactly those reads. And now we are going into Metagenome Assembly for one step, because we'll need that. We'll need those results later on. So this is one way of doing metagenome assembly, where you're really interested in a specific in a specific species, sort of dragging out those reads from a metagenomic sample, and then looking into which species specifically is present here. Let me first save all those reads that are associated with that species, which is SARS-CoV-2 in this case. So we have these filtered reads, and then we can say the Denovo Assembly select those, keep all the parameters at default. We'll choose this fast route, and then we'll see. Oh, this is already done. Briefly look into it, and then we can see, okay, there are a few bigger contigs and some smaller ones, but all in all 14 contigs. As I said, we'll later analyze those a bit further. Let's first go back to the abundance table, because it is of interest how to analyze such a table in the context of metadata. So the first thing we need to do with respect to the abundance table is that we collect all of the abundance tables into a single abundance table. So the output from TextPro would look like this. As I said, all one folder per reference per read sample. So, and we have one abundance table per sample. What we need to do is we would need to run a tool that is called merge abundance table. And then to make it a little bit easier, and we don't want to open each of these folders individually, we can again right click on the TextPro folder here and then select add folder contents recursively. So it will go into each of the subfolders and check whether they're fitting data elements. Then we have added them all, and we choose to save search in abundance table. So now we have an element that is called merged. If we look into that one, it looks a little bit similar to the one before, but it has many more columns now, one for each of the samples. Also, what is possible now is that we can aggregate those samples based on the metadata. One problem we have is that the metadata here is not the metadata we're interested in, but what we really want is the metadata from the inline supplementary material. So in order to associate such external metadata, we have a tool that is called add metadata to abundance table, which we will just run on this merged element. Now you can see, because I had selected it here, it's already selected in the navigation area here. Click next. In this case, I have already browsed and selected it. We need to make sure that one column here is called name, and that column should correspond to the names of our samples. And once that is done, we can click next, click save, finish. Now let's see. You have briefly seen something blinking, and if you are unsure whether the process has been successful, well, it didn't tell me it was unsuccessful, at least. And I can also look into, by clicking on this small triangle here, looking into the log information, and then it tells me that it associated metadata for 11 samples of 11 samples. So all of these samples should have the new metadata associated now. And it's rather simple to see that now there is additional metadata in here that is written in non-capital letters, and that's the one that's coming from the external spreadsheet. Here it would be interesting to compare different locations, because they have taken the samples at different sewage plants, and then we can and then if you select to aggregate on the location, then you can see the headers of the table change, and it actually tells me how much of each of these was present in the different locales. But there are different ways to visualize this. We also have a we also have a stacked bar chart visualization in the bottom here. Oh, this is a little bit weird. Let me see if I can stop this process. This is probably something that is doing this. Yes. And and the stacked bar chart will give you the species composition at a given level of the taxonomy, which you can also choose and select for all of the samples. You can, for example, sort the samples according to the metadata, and here we would like to sort them by the location. Then we can see how many samples are associated with Oakland, Berkeley, and Marin. But you can also choose to aggregate the samples based on the metadata in the same way. Date, location. So furthermore, there is a Sunburst editor which lets you explore the same data in a little bit different way. But again, you can choose to aggregate the sample based on, for example, location, and it will always show the combined abundance here. But we can also inspect them individually, either for Oakland or Berkeley, or Oakland and Berkeley combined. And you can also increase the level of resolution here to see what is going on further down. Well, yeah, that is interesting, but not really a statistical analysis yet. So we have, as I said, a range of tools that can be used for the statistical analysis of such abundance tables that can be found in metagenomics abundance analysis. And you can choose, for example, to create alpha diversity, beta diversity plots, and Terminova analysis, create heat maps for your samples, and cluster your samples and your taxa respectively. Let's have a brief look at the alpha diversity. And I would like to get the alpha and beta diversities on a species level. So I can either do that here or later, but I'll choose to do that here. Aggregate my features on species, say all, and I select all of the rows and create an abundance subtable that is now filtered and collected on species level. For this one, I would like to run the alpha diversity tool. And I'm now, for now, only interested in the total number. As I said, you can also choose to aggregate here, for example, on species level or any other level. The rest we keep as default and finish. While this is running, let's also briefly start the beta diversity, which is much faster. Let's choose one of them for now, and we'll save this here. While alpha diversity is running, let's have a brief look into the beta diversity. So, oh yes, yeah, you can see now we have only three samples left. That was because I selected to aggregate the features aggregate the samples on the location. That was not my intention, but you can see that this is also doable. So let's for now aggregate on species level. Use the subtable and save it here. Great. Alpha diversity. Next species. Let's have a few more points. And create the beta diversity again. So close all of the other ones, and then we can, for example, look into our samples by beta diversity. Also here, if they would cluster, you would be able to inspect it by coloring, either by categorical variables or by abundance variables. For now, let's have a look at our location variable and see if sort of the locations end up in the same corner of the principal coordinates components plot. And it does not really seem like it's clustering well. Also, I mean, there are not too many replicates of some of the samples like Marin here in Berkeley. but there is some difference. So also note that these plots are available in 2D, and sometimes you can actually see signals in those. One other type of analysis while the alpha diversity is running is the differential abundance analysis, where you can choose an abundance table and basically compare groups of samples. For example, again, we select the metadata table. We would like to analyze it with respect to whether there are differences in the related to the location of our sample with the location metadata. Where is it? Here it is, location. We could also correct for confounding factors. We'll not do that for now. There are different ways to run this analysis, either across all of the groups, where you basically lose the group information, but it will tell you, is there something differentially abundant across those groups? We can compare all the group pairs, or we can choose to only use one control group, where we use all of the groups that we use against one control group. Let's do the all group pairs analysis for now. Open it. And then we see, okay, we get a very big table with, for example, Berkeley versus Oakland, Marin versus Berkeley, and Marin versus Oakland. So all of the group pairs. For all of these different pairs, I get the different taxonomies listed. And for each of the species or higher order taxonomies, if they don't have a specified species level, I can see whether there are changes when between Berkeley and Oakland, for example, and then I can also see whether these the log2 fold changes, the fold changes, and whether they are significant with p-values and FTR and Bonferroni corrected p-values. Furthermore, these group comparisons can be looked at in a Venn diagram view by control clicking on this Venn diagram. It will open in a split view, and then you can select rows from the above, for example, by clicking on each of the segments. And note that the numbers here represent the number of significantly different taxa across those groups as defined by the FDR corrected p-value with a significance level of 0.005. But all of this can be controlled basically according to how much you trust your data. In this way, it lets you identify differentially abundant species or OTUs or differentially abundant functions across metagenomes. So these abundance analysis tools, they are very general and can be used for all different types of abundance tables. So now also our alpha diversity process has finished. We can see it is at 100%. This is the result. We can either, because I know it looks like that, but you can also, if you don't know how it looks, you can also, well, you can't. You should be able to show results. I'm not sure why this is not possible here. Well, if we look at it, then we get these alpha diversity curves, and we can see that, oh, it actually looks like we capture most of the species, so it has been sequenced quite deep, which is good. And now we would like to see whether differences between the groups again. For this, there are we have an alpha diversity box plot here, which takes different, where you can look at the data at different rarefaction levels, but only rarefaction levels that are present in all of your samples. So if you only have one available here, then it's then you either need to increase the number of points, or you need to remove some of your samples. Well, now this looks a little bit dull, because for each of the samples there is only one point, of course. So the first thing we would like to do is we would like to group these samples again by metadata. You can see that under the group by, there is again our favorite location setting, and then we can see that there are some samples for Oakland, one for Marin, and two for Berkeley. And then we see that they have different total number of species detected. Whether there are significant differences that can be evaluated by looking at the p-values, which are displayed above. So a Kruskal-Wallis p-value across all of the samples, and specific Mann-Whitney U-test p-values, where you can choose a reference or where you can choose to show all of the p-values. So there may be a somewhat significant difference between Berkeley, the diversity in Berkeley, and the diversity in Oakland. So with this, I would like to finish the analysis of abundance tables or the statistical analysis of the abundance tables we feature, and get back to the metagenomic assembly we have been doing. So remember our context here. Now we would like to see whether these contexts are really from SARS-CoV-2. So remember, we have pulled out the reads that map to SARS-CoV-2 with the taxonomic profiling tool and assembled them into context. Now we would like to see how they match. For this, we have installed one way to go about this is to use the whole genome alignment plugin, where you can either create dot plots or create a whole genome alignment. I'll start by creating a whole genome alignment because I think the tool has some strengths. For example, it lets you select a reference genome, which I choose as the SARS-CoV-2 Wuhan reference, and this reference comes annotated with CDS, and I can choose to copy annotations from the reference to my context now. I will also keep the rest as is here and choose to open. So here I can see, oh yeah, this seems to align pretty well. And if I now choose to look at the annotations, I can also see that the annotations have been transferred from my reference genome, indicated by this little star here, to the other genomes. This is quite a boring whole genome alignment, but let me point out that the number of genomes you can put in here it can be a lot larger, and then also the whole genome alignments look a little bit more interesting. With such a whole genome alignment, we can even inspect how good our assembly has been. For example, we can create average nucleotide identity plots for this. If we don't select any annotation types, it will be done over the whole genome. And then we can see the alignment percentage below. So we have 92% of the reference covered. MN90, so this MN sequence is our reference, right? And in those 92%, we have a 99.94% identity, which is actually quite good for a metagenomic assembly, or with reads that have been extracted from a metagenomic sample. If you want to do a full metagenomic assembly without going through the detour of running taxonomic profiling and dragging out the reads, we have a tool that is called Denovo Assembly Metagenome. But in this case, you would of course expect that your contexts are not species specific, but across many species. So you would have to associate taxonomies to those contexts after assembly, This tool takes simply trimmed reads as input, and otherwise you can run it as any other tool. For example, let's do this here. We would choose longer contigs and save. Now remember, we have just used the metagenomic contigs and not the SARS-CoV-2 specific ones, but this is just straightforward. So after assembly, you would then have to classify those contigs. We have tools for that. I will not go into detail, but there's a plethora of options. For example, binning the contigs with these two tools, either by sequence similarity or by taxonomic identity using a reference database. And then you would possibly, as a next step, associate it with CDS, either by aligning it to their respective references using the whole genome alignment tools, or using the annotate with BLAST or DIAMOND tools and a respective BLAST or DIAMOND reference database, which again is obtainable in the databases subfolder. Last but not least, I would briefly like to mention that if you have amplicon reads, the analysis steps are very similar to the taxonomic analysis we have just run, only that your entry point is a bit different. So when you go to metagenomics, there's this assembly-based analysis button. And here we have a tool that is called OTU Clustering, which you would run instead of taxonomic profiling. This OTU clustering, let me briefly show you, gives you also an abundance table on output. And this abundance table is very similar to the taxonomic abundance table from taxonomic profiling. And it can do all the same things in terms of visualization and downstream analysis. So 16S data and ITS data is analyzed in a very similar way, only that the tool provides some different options compared to the taxonomic profiling tool. With this, I would like to conclude today's session and thank you very much for your attention. I would really be happy if you took the leap of faith and tried our software. I think it's very easy to use. It's cross-platform. It works both on Linux, Windows, and Mac, and it's easy to install and easy to use. Other than that, we're happy to get feature suggestions if you have them. Please reach out to us whenever you feel like it. And thank you very much for your attention. Goodbye.